when I came to U of I and I took like the first introductory biology class, it was like, okay, this is basically like another world, you know, and I want to know about it because it's a future.
Yeah, it's the future. What do you mean by that? Elaborate on that.
Well, to start with, like CRISPR, which was, I will say recently discovered. It was, yeah, back in the 2000s. Like CRISPR right now is the future. We can talk about it later. Like, but there's so many applications on that.
Why don't we get into it right now? Describe
Okay.
for listeners and viewers, what is CRISPR?
Well, CRISPR in the most simple terms here is a tool for gene editing. I think you can have with it many applications in the medical system, such as for people are right now trying to use it to cure syndromes like Down syndrome, maybe Alzheimer's. Also, they're using it in crop sciences. And actually, I just saw a study like probably a couple of weeks ago where some people were trying actually to cure blindness with CRISPR.
So these are some big challenges that this is potentially can solve. What exactly is CRISPR? Is it a technology? Is it a stem cell, or what exactly is it?
Well, so if we go back a little bit in time, CRISPR is something found in bacteria. The most common meaning of a bacteria is a bacteriophage. And CRISPR, it was basically a system localized inside the bacterial DNA that helped to combat the bacteriophages. So it was like a defense mechanism.
So is CRISPR like an actual biological thing, or is it a technology?
No, it's actually a thing that happens in bacteria, and they use it to defend against bacteriophages, which is a kind of viruses which infect bacteria.
Okay, so how does it work?
CRISPR? Okay, so if we want to talk a little bit about biology here.
Please do.
Okay, so CRISPR. Okay, so we're gonna go about this. We have a DNA sequence, right? Let's imagine we have a DNA sequence of a bacteria, right?
Yeah.
And a bacteriophage comes and they're like, oh, I like this bacteria. I'm just gonna insert my RNA here. The genome of the bacteria has already in like his system, like saved that sequence that the bacteriophage is infecting to the bacteria. So as soon as the bacteria detects that sequence, he's gonna synthesize this protein called the Cas9. The Cas9 has a helicase activity, and by that I mean that it can cut through the genome. So the bacteriophage is gonna come, is gonna infect the bacteria with that RNA. The bacteria is gonna synthesize the Cas9, and the Cas9 is just gonna destroy the genome.
So so it's gene editing, right?
Yes.
What makes it? Yeah, go ahead.
So yeah, when it comes to gene editing, so there's like another mechanisms involved in CRISPR. For example, is gRNA. And when it comes to gene editing, like in research, what we use is a sgRNA. The sgRNA is something that is like a sequence of probably 20 nucleotides long that is gonna pair up with a sequence of your interest. And the sgRNA works as a guide for the Cas9 to go like, oh, here you are, I'm just gonna cut here.
So what makes it different than, say, like we've heard terms like genetically modified foods or seeds, or like what makes this technology or breakthrough different than before when we've been able to like edit genes in a way or manipulate them in a way?
Well, this technology actually is quite cheap. As like, actually the doctor who discovered CRISPR, I think her name is Jennifer Doudna. She said, yeah, CRISPR compared to other gene editing tools, this one is way cheaper and way more effective.
So that's the big thing, is like it's astronomically cheaper than
Yes.
everything else.
Yes. It's way cheaper, and I feel that I think it's quite easier to use if you know like all the theory behind it, because each organism is different. But
Okay.
whatever with which organism you want to work with, like you gotta know how to apply for it.
So is it the same? So has this so the reason it's let me think. So this is like a really big deal in the science community.
Yes.
Is it because it's just that much cheaper than everything else? Is that the big thing, or?
Yeah, it's like because like mostly everyone can get their hands on it, to be honest. And there is where it comes to bioethics, because like I remember like a while ago that I think there was this Chinese doctor called Dr. He, who was able to actually modify the whole organism of a fetus back in China.
Mm-hmm.
And it involved such a big controversy with CRISPR.
Yeah, that's okay. So what are okay? So to summarize, CRISPR it's a gene editing tool.
Yes.
That we can apply and take out certain DNA that creates this or that.
Yes.
And we can do it much cheaper. That it makes us it allows us, it's so cheap that it allows us to do it.
Yes. And it also works like to knock out genes specifically. There's like editing genes and knocking out the gene. Knocking out the gene is like basically shutting it off. So it's no longer functional.